Μπορούμε να προχωρήσουμε στη δεύτερη διάλεξη του καθηγητή Άντρεα Λεωνάρντι. No words to describe. Αναπληρωτής καθηγητής στο Πανεπιστήμιο της Πάντοβα στην Ιταλία, στο τμήμα της νευροοφθαλμολογίας. Η διάλεξή του έχει θέμα Tissue Remodeling in Ocular Allergy. Professor Leonardi, hello from Greece.
Hello, how are you doing? Thank you for the invitation. I would like also to say hello to Manta, the friend of us. Thank you.
Hi, Dr. Leonardi. Pleasure meeting you here.
Thanks. It's too bad we cannot see each other in Salonico, but it's a pleasure to be virtually.
Even
there.
visually, yes.
Thanks.
Good afternoon. I would like to thank the Atlantic Society for the study of ocular inflammation and infection for this nice invitation. Today we'll talk about tissue remodeling and ocular allergy, a very specific topic in ocular allergy in particular. Ocular allergy is one of the major ocular surface disorders affecting million people. It goes from different mild forms, usually associated with rhinitis, to severe forms of like VKC, KC that might cause several problems to our patients. Ocular allergy can be divided in IgE and non-IgE mediated diseases. In particular, the allergic response is a type 2 mediated immune response with Th2 cells involved, which might stimulate B cells to produce specific IgE or differentiate the Th2 type effector cells, which in combination might cause acute and chronic inflammation, in particular in conjunctival tissue. There are several aspects of allergic inflammation that might be taken in consideration as cofactors in your left and as effects on the ocular surface. Today in particular, I will talk about the tissue remodeling and briefly epithelial dysfunction. Tissue remodeling can be classified as a persistent changes of the conjunctival structure involving the composition, organization, and functions of structural cells, as well as enhanced turnover of extracellular matrix components. This is particularly evident in VKC. VKC is a persistent and severe form of ocular allergy that affects children and young adults, usually in warm climates. In fact, the grading of severity of VKC takes into account both limbal changes and the size of the forms of giant papilla in the upper tarsal conjunctiva. Talking about tissue remodeling, I will consider these three different aspects, some histological studies we've done in the past, fibroblast response, and some idea on gene expression. This is the three-dimensional aspect of the giant papilla, in this case a single giant papilla in the upper tarsal conjunctiva, and the histological picture with intense mixed inflammatory cells infiltration. The remodeling involves the conjunctival epithelium. There are several different morphological changes that involve the epithelial cells, hyperplasia, epithelial ingrowth with formation of glandular or pseudoglandular hypertrophy, global cells hyperplasia, and as a consequence of that, and mucus hyperproduction. The stroma is also involved with the increased deposition of collagens, in particular collagen 1 here in red, which is not only on the core giant papilla, but forms like a layer in the subepithelium. There are other collagen extracellular matrix components that are increased, in particular VKC. We and others describe increased production and deposition of collagen 1, 3, 4, 5, and 7, and 7 other extracellular matrix components such as fibronectin, laminin, tenosine, periostin, and relative reduction of the glycosaminoglycans. These are some of the proof of this increased expression of tenosine, laminin, and fibronectin. This extracellular matrix component facilitates inflammatory cell migration into the conjunctiva and also the cornea in some cases, and increases the activity of metalloproteases. The question is tissue remodeling an inflammatory effect as a result of repairing mechanism, or is it an independent phenomena? We really don't have the answer to this question. However, we know that the epithelial cells, inflammatory cells, fibroblasts, and extracellular matrix have a crosstalk each other, and might represent the epithelial mesenchymal trophic unit, so a functional unit that regulates the physiological processes. We know that the fibroblasts derived from VKC patients show a faster growth in vitro and are more responsive to cytokines in terms of collagen production compared to normal fibroblasts. So it means that might be some intrinsic factor that might stimulate the faster growth of these cells in vitro. In fact, the Th2 cytokines, particularly interleukin-4 and interleukin-13, stimulate fibroblasts for production and expression of collagen 1. On the other side, interferon gamma reduces the production of collagen 1. But it's not only cytokines. We found using multiple assay of factors in VKC that several growth factors are also overproduced, at least in VKC. Of these growth factors, TGF-beta1 is definitely the most important one related to collagen production, but also we found increased expression of PDGF, FGF, and VEGF as well. These factors are produced by both resident cells, meaning fibroblasts and part epithelial cells, but also by inflammatory cells. This is a particular form of VKC-like hypereosinophilic syndrome in a patient with a gene defect in FIP1L1 PDGF receptor alpha, which give the proof that PDGF is involved in tissue remodeling released by eosinophils. Several of these growth factors are overproduced, in particular TGF-beta1, which is highly expressed by inflammatory cells, in particular eosinophils and monocytes and macrophages. We have shown that not only TGF-beta1, but also TGF-beta2 and the whole cascade of SMAD intracellular signaling pathway are overexpressed in VKC, showing that the whole cascade of events involved in inflammation, tissue remodeling are present in this disease. So to conclude this part of my talk, there's the remodeling in VKC is characterized by increased deposition of collagens and extracellular matrix components, increased expression and production of growth factors, and increased responsiveness of VKC fibroblasts to different factors, particularly Th2 type cytokines. So beside the cytokines, the growth factors, there are also overexpression. There is also overexpression of several metalloproteases, particularly in VKC, as shown by this multi-well assay using tears from VKC patient. You can see in the first panel here, several metalloproteases are overexpressed in VKC compared to control subjects. These are metalloproteases and the two inhibitors, TIMP1 and TIMP2, which are normally expressed on the ocular surface. Not only proteases, but also the antiproteases or protease inhibitors are involved. We have shown that there is a reduced activity in alpha-1 antitrypsin, which is the archetype of serine proteases inhibitor in different tissues. And we also found relatively low levels of proteases inhibitors, TIMP1, PI1, and SERPIN1, which is also downregulated, particularly in VKC. So for this part of my talk, the proteases are taking place in the remodeling process because they degrade the collagens, they alter the function of cellular matrix components, they facilitate its cell migration and acceleration, modulate cell-cell interaction and cell matrix interaction, but also release and activate several growth factors and mediators that are trapped in the tissue. Then we compare tissue remodeling in different allergic diseases, particularly VKC, polyps, nasal polyps, in asthma, and found that actually there were similarities, but also differences in collagen deposition between VKC, polyps, and asthma. So it was similar in VKC and polyps, but in asthma, the deposition of collagen was completely different, like a monolayer under the epithelium. Only differences in collagens, but differences also in metalloproteases expression in VKC compared to polyps and asthma, as is shown in this small table. Sorry for the size, but I just want to highlight it, that in VKC there was the highest production and expression of metalloproteases compared to other tissues. This is not just the well-known MMP9, but also other MMPs were overexpressed. But also the number of inflammatory cells present in the tissue was much higher in VKC compared to polyps and asthma in terms of both eosinophils and Th2 cells. The second aspect we would like to touch is the fibroblast response. Over the years, we've been looking at different stimulation of fibroblasts, starting from histamine. Histamine is the main mediators released by the allergic response. We have shown that histamine through H1 and H2 receptors stimulates fibroblasts in production of different collagens and therefore induce fibrosis. Not only histamine, but also interleukin-4, 13, and TGF-beta-1, which are overexpressed in tissue, increase the expression and production of collagen-1, but also they increase the production of TGF-beta-1 and TGF-beta-2, therefore increase the loop in the collagen hyperproduction. We also know that there is a very close interaction between fibroblasts and inflammatory cells, particularly eosinophils, because fibroblasts are the highest producers of eutaxin 1 and 2. Eutaxin is the strongest chemokine for eosinophils, and on the other side, eosinophils produce high levels of TGF-beta-1, which is the main stimulator for the remodeling processes. A few words on gene expression. Lately, we have been in a different gene expression again in VKC. So this will be actually the topic of my second talk in this meeting. I just would like to highlight the fact that in tarsal VKC, one of the highest overexpressed gene is TGF-beta1. Again, showing that remodeling process is highly activated in the subtype, this sub form of VKC. Doing another gene expression using the Affymetric technology, looking for a high number of genes, we can see, these are not published data, that in limbo VKC, the most gene ontology biological processes expressed were related to ribonucleoprotein complex biogenesis. On the other side, in tarsal VKC, the most families that were overexpressed in terms of gene expression were related to tissue remodeling. We also have been interested in the epithelial barrier function and factors that are related to the function of epithelium and the interaction between epithelium and the subepithelia, which is can be modulated and modified in the epithelium-mesenchymal transition, which is a modification that might occur in chronic inflammation. So we found that there are several genes overexpressed that are related to the tight junction and the relation between epithelial cell to the adjacent epithelial, but there are some downregulation of other factors. We believe that the epithelium-mesenchymal transition is not present, at least in VKC. However, also we can tell there is a remodeling process going on inside the epithelium as well. So in conclusion, what can be called the epithelium-mesenchymal trophic unit that regulates the epithelial barrier inflammation and remodeling induce kind of vicious circle on the ocular surface, modifying the barrier function into a barrier dysfunction, increasing inflammation, and inducing tissue remodeling. I would like to acknowledge several collaborations I have and had in the past with centers and researchers that helped me in these findings, and would like to thank you for the attention.
Thank you very much. Υπάρχουν ερωτήσεις στο κοινό για την ομιλία του κυρίου Λεωνάρντο? Leonardi?
For Mr. Leonardo.
Leonardi, may I ask you something? I don't know how much is related with that. Is there a clinical test in your clinical practice for evaluating the ocular surface condition in allergic conjunctival diseases? In your clinical practice?
Thank you. My presentation was showing a lot of data that are related to preclinical observation. Of course, into clinical practice, the first thing we do is looking at the patient and talk to the parents in case of children, and evaluate the ocular surface with the slit lamp. So there's no particular test that will tell you what is going on the ocular surface beside the clinical observation, at least in the case of ocular allergy. Of course, we can use additional multiple tools from tear cytology, confocal microscopy, and tear evaluation for biomarkers. But at this point, we have a single biomarker to be used in clinical practice. This is kind of frustrating because after a lot of measurement and trying to find, you know, a tool that might help practitioner in everyday evaluation, we still don't have a useful tool to help clinicians.
Thank you very much.